All righty, welcome everyone. So today we're going to be talking about single-cell genomics. On Tuesday, we basically talked about the gene expression analysis, and then I told you, oh, and there's a lot of complications with single-cell genomics, and we're going to talk about them in lecture 13. Well, guess what? We're going to talk about them today because your problem set has basically the entire pipeline of single-cell analysis, differential analysis, pathway enrichments, some single-cell epigenomics, all blended in an awesome Jupyter notebook. Why are we doing this? So that as you start thinking about your final projects, you have all of the tools in your disposal. And a lot of people have talked about single-cell genomics in their projects. So we want to enable and empower all of you guys to be able to actually go and carry out all of these single-cell analysis, which are frankly a lot of the frontiers of where biology is right now. So a lot of mysteries of how the genome works and how disease impacts the body is very difficult to uncover when you're looking at bulk analysis. However, the moment you're able to separate the different cell types in the body, things become much more amenable to these types of analysis. So let's dive right in. So we're going to basically now start with why single cells. And hold on a second, make sure that everything is here. Good. Go. Why single cells? Basically, what are the traditional approaches and sort of the emergence of single-cell RNA sequencing across different types of technologies? Because understanding the technological underpinnings can also help you understand some of the issues with noise and when the technology doesn't go exactly right. Then we're actually going to dive into the biology first. What are the questions that you can ask about single cells? And we're going to focus on a lot of the work that our lab is doing on understanding the brain in the context of Alzheimer's, in context of schizophrenia, and so on and so forth. Then we're going to step back and say, okay, what are the basic computational tasks associated with single-cell analysis? And then we're going to look at some of the frontiers of the field of some deep learning, representation learning methods for understanding single cells. And then we're going to touch briefly into beyond transcriptomics, into some of the epigenomics, multiomics, and so on and so forth. Who's excited? Yes? Awesome. Great. Let's do it. So why single cell? Again, there's many ways. Oh, yeah, go ahead.
I don't think the slide is locked. Oh.
Like on campus. Yeah.
We'll release it after lecture.
Yeah, they're slowly, you know what? Here. That's why I always bring a thumb drive, because it starts syncing up there and then it continues syncing until, you know, the end of days, or at least the end of lectures. There's also PDF being generated, so the PDF might actually appear at some point, but we'll see. Okay. I love how there used to be a time when the teacher would basically just write on the board and you would sort of see what the next sentence is when it's written. And now there's like, oh gosh, the slides are not yet online. What are we going to do? So it's, you should be expecting more. And that's the progress of technology. And that's, you know, we should always be asking for, any kind of information that can be shared, should be shared. And that's why we're recording the lectures, for example. You know, there's students on Harvard who are like, you know, back to back, unable to come. They're able to watch them online. And the first lecture got, what is it? 1,000 views or something like that, or 4,000. Like, anyway, I posted it on YouTube, and there's like thousands of people who are watching it and sort of talking about it, which is great. So it's kind of cool. But anyway, you guys are the originals. This is great. This is you get a 2-hour head start on sort of the frontiers of research. Okay, so there's many ways to think about gene expression analysis. The easiest, and the analogy that has been used in many talks and lectures and newspapers articles, is the smoothie versus the individual fruits versus the fruit salad or the tart. So when you're doing bulk transcriptomics, when you're basically taking a piece of liver and profiling it, remember in the first lecture, or Tuesday's lecture, I was basically saying, oh, if you look at brain expression versus liver expression, it sounds reasonable, right? Like if I tell my parents, oh yeah, we looked at brain expression versus liver expression, it makes complete sense to them. But to a scientist, and especially somebody working on liver or brain, they're like, liver? Okay, which of the 47 cell types in liver? And somebody working on brain is like, which of the 143 subtypes of, excitatory neurons in the brain, et cetera. So basically, when you think about liver expression, what you basically do is that you take this extraordinary diversity of cell types and you blend them together in a beautiful smoothie. And then you're looking at the liver smoothie or the brain smoothie or the lung smoothie, et cetera. When in fact, it's a terrible analogy. When in fact, the individual fruits are basically what you're after. And what we're going to look at today, especially in the context of self-projected phenotypes, a measure that and a method that we've been developing in my group with Gerard Bouland and others, is that even within cell types, there are continuums of variation that are very often biologically driven. So even at the level of sorting the beautiful kiwis, there's no such thing as like one type of kiwi. There's still a gradient of kiwi, and they're associated with different biological properties, sometimes different disease properties, and so on and so forth. So when our group started looking at single cell biology, it was very, very difficult to go back. And we've kind of gone all out, and then we've profiled more single cells than we could have imagined. And that's what I'm here to share with you today, that basically, if you look at sort of the number of cells that people have been profiling, it's now in the millions. And from my group alone, we have more than 20 million cells that we have profiled, which sounds unfathomable. Basically, if somebody told you, oh, I'm going to, give you 20, like, remember when I showed you the gene expression by condition, it was like thousands of genes, hundreds of conditions. Now we're not talking about hundreds of conditions or thousands. We're talking about not tens of thousands, but like millions and tens of millions of conditions, which are the cells. Okay. So that's why it's important, because in the brain or in the lung or in the liver or in every cell type in every tissue in your human body, there's an extraordinary diversity of interplaying cell types. And that's concept number one. Concept number two is that very often we're going to be talking about single cell, but in the brain, the neurons are sometimes extending all the way out to our toes, okay? So how do you do single cell profiling? You're going to have to sort of pull out all of that neuron, take all of the RNA from all of the different axons and dendrites and sort of, unweave it and then profile it. That's very hard. And especially in the brain where the cell types, including oligodendrocytes, astrocytes, and of course, excitatory and inhibitory neurons, they're extraordinarily interweaved with each other. So it's very difficult to sort of pull up all of the parts and then sequence the entire content of the RNA of one cell. So an approximation that we're nearly always doing is single nucleus sequencing, because the RNA that's in the cytoplasm at some point needs to be produced from the genome, and that is done in the nucleus. So you can think of it as an approximation of what's being produced right now, rather than the actual actin molecules that are sort of producing proteins in the cytoplasm of these cells. Moreover, as we start thinking about more and more resolution, I wouldn't even want to know what's the total content of RNA inside a single neuron. What I would like to know is how much are sitting in this part of the axon, in that part of the axon, in these dendrites, in those dendrites, et cetera. In other words, the transport of RNA along the cell body to the different parts is also extraordinarily important. And I think that's also a technological frontier. So there are some purists that will basically say, oh, single nucleus makes no sense. Why are you doing this? And there's other purists that will say, RNA expression makes no sense. Everything happens at the protein level. And other purists that will say, protein makes no sense. Everything happens at the post-translational modifications of the protein level. And others will say, oh, post-translational modification of the protein makes no sense. Everything happens at the localization of the post-translational modifications and the orientation of them, et cetera, et cetera, et cetera, et cetera. So my answer to all of those purists is, we got to start somewhere. And we start where the data is. We start with the technologies. We start with what's accessible right now. And based on what we learn, we help advance the field and the technologies. And we continue pushing the frontiers of single cell profiling to more and more different types of modalities and technologies and so on and so forth. that will not be possible if we didn't push every technology to its extreme based on what's available today. In other words, yes, we could have waited 10 years back in 1995 to basically say, okay, single molecule sequencing is around the corner, and long read sequencing is around the corner, and sequencing is very expensive right now. Why sequence the entire human genome now? Let's wait, a decade or two for the technology to improve. But the catch is that the technology will not improve unless you do it with the current technology to show the usefulness, to drive innovation, to drive entrepreneurship, to drive technological advances, to drive the market and the need for these advances that will then feed back. Everybody with me here? So that's why we keep pushing, even though we know it's an approximation. So again, single nucleus RNA sequencing is extraordinarily awesome, gives us a ton of insights, but we should all be cognizant that there will be more as more technologies become available. But until they are, we're going to keep pushing the frontiers of what is available. Sounds like a plan? Awesome. So again, I talked about the smoothie, I talked about the individual fruits, and then there's the tart, which is the third analogy of spatial transcriptomics, which we'll allude to a little bit here, but then we'll also talk more when we start talking about imaging analysis and so on and so forth. And that's basically when you look at the tissue itself, where every single one of those cells is positioned, rather than only looking at sort of these cells pulled out of their context. So basically, the single cell technology, you should realize, is extremely limited because it tells you about, yes, all of the excitatory neuron layer 2, 3, and so on and so forth, but it doesn't tell you exactly where these are positioned. And maybe the ones that are positioned in this part or that part or associated with, I don't know, vasculature or not, are actually different from each other. And that you can get from spatial transcriptomics. Everybody with me here? Awesome. So, great. So why single cells? I think I got a lot into that. And, let's talk a little bit more about this. So individual cells, again, what I've been saying is why single cell types, why studying the kiwi separately from the banana is extremely important. But what's also important is studying multiple kiwis separately and multiple bananas separately. And the reason is that when you look at cells, in the body or in a plate, they actually look very different from each other. And when you look at them in a microscope, they have different morphologies, they have different expression levels of individual proteins, and one cell to the other, even of the same cell type, can be dramatically different. Okay? Moreover, if you look at the blood differentiation, you have extraordinary diversity and almost a continuum of cellular identity as you go from the hematopoietic stem cells, which are the stem cell progenitors of all of the different blood lineages, the hematopoietic lineages. And then you have this extraordinary diversity of cell types that we know have different functions. And then, of course, there's the diversity within the cell types themselves of each of the different functions. So all of this needs to be taken into account. Moreover, why are they different? They're different because of stochasticity. So basically, every cell has an expression program, but which subset of genes gets activated at any one point actually differs. So based on the genes that get activated first, even though all of them are supposed to be turned on, you might get some that get turned on first and others second, etc. And some of that is stochastic. It's how will the particular regulator wash through, quote unquote, the different regions of the nucleus and open up different parts of the genome to express different genes. There's also stochasticity in how a signal that is intracellular will permeate a group of cells and how these cells will respond. So the receptors of a particular cell might receive more or less of a given environmental signal that then causes a cellular state transition. And that can actually lead to a lot of the diversity even between individual cells. And then lastly, developmentally, every cell has a program that starts from a single genome, and then through series of divisions and maternal deposition of gradients, and also where a particular cell is relative to, the site of implantation or you name it, all of that will lead to changes that happen at every cell division that basically say all of, this lineage after this one cell division will be ventral. and all of these lineage will be dorsal, or all of these lineage will be anterior, and all of that lineage will be posterior. And there's series of decisions that get made after every one of those divisions that basically lead to different identities, which are remembered by the epigenome that we're going to talk about next week. They're remembered by the epigenome, and then they determine the responses of each of those cells to subsequent signals. Is everybody with me here? Yes? Okay. Can I get a 5, 4, 3, 2, 1 for how well you're following? Let's see some 5s. This is great. Awesome. Some 4s. Any questions? Who feels like they're learning stuff? Yes? Awesome. Great. Beautiful. Okay. So when you start looking at individual cells, and some of the initial methods were basically taking a tissue, dissociating individual cells, and then pipetting these cells and putting each of these cells in a separate well, and then amplifying the RNA of that well, and then sequencing that one well. Okay? This is the original single cell profiling. And then you got like 600 cells, and it was extraordinary. And what you could see is how these cells can be dramatically different from each other. So you can distinguish different types of cells, different responses, different stages of activation of those cells after 1 hour, after 2 hours, after 4 hours, after 6 hours of a particular stimulation. You can kind of see which subset of cells is responding in which way, and what subset of genes are activated in response. So this is the expression matrix that I showed you in last time's lecture. Everybody with me here? So what we're showing now is not hundreds of conditions, but instead hundreds of cells, all living in the same sort of organ, or in the same, sometimes animal or donor. Okay? So that's the traditional approach of isolating individual cells using a pipette. Then there's automated approaches for doing this, where you're basically shooting cells through cell sorting apparatus that allows you to now start, putting each of these individual cells in wells. And there are technologies that allow you to now encapsulate every cell in a microfluidic droplet that allows you to now capture individual cells in reaction chambers that are not the size of a test tube, that are not the size of a plate with 196 wells or something, but is instead the size of roughly one cell, where basically those reaction chambers are made out of little capsules, little droplets that are encapsulating those cells. In every one of those droplets, you can put a bead that contains millions of barcodes that will then tag every single one of these RNA molecules with the same barcode. And then you can sequence the resulting RNA and look at the barcodes and then try to infer which RNAs came from the same capsule, from the same droplet, and therefore which RNAs came most likely from the same cell. Everybody with me here? And of course, when we're isolating individual cells, or when we're isolating individual nuclei, there might be stuff stuck on them. And as we lyse the nucleus, or as we lyse the cell, there might be some ambient contamination of those cells. And that contamination might be greater with the health of the original cell. If you're taking cells from a cancer patient, they might actually be more prone to this type of contamination. If you're taking cells from a very elderly individual, or from someone with neurodegeneration as the neurons are dying, the state of their nuclei, the state of their cells might actually be compromised. Not just when you're carrying out your experiment, but even before. Maybe there's more types of contamination that happen, because that individual is sick or elderly and so on and so forth. Yes?
Are these RNA molecules pre-mRNA, or are they spliced on these sequence?
So because we're looking at the nucleus, it's both. And you can basically get at some of the dynamics of gene expression patterns by asking what fraction of my mRNA molecules are actually unspliced versus spliced. And there are techniques for building cellular trajectories. In a differentiation, for example, if you think that all of these cells are part of the same differentiation lineage, leading from state A to state B, and between state A and state B, I need to express, I don't know, 40 genes, you could basically say, oh, let's find cells where this gene is now fully spliced versus still unspliced, etc. So based on the coverage of the splicing of the introns in between the exons of every one of those cells, you can actually tell a lot more by exploiting the nature of the data set. But yes, the answer is they're going to be both spliced and unspliced because you're capturing nuclear RNA in many of those cases. Everybody with me here? Awesome. Good. So again, if you start looking at bulk and there's many cells that have little and many cells that have a lot, you're going to mistakenly think that, your tissue is somewhere in the middle, when in fact, very, very few cells are actually there in the middle. Similarly, if you have some rare cells that express a lot of a given RNA, and then you average the whole tissue, you're going to think that, oh, every one of those cells has a little, when in fact, most have none, and a few cells have a lot. Okay? So these are some of the reasons. And then this is now looking at multiple genes on the x-axis. So here you can see the actual exon intron structure. So as you all know, there's the original genome here, and then every tick mark is an exon, a protein coding exon. And then these exons get spliced as they get transcribed. There's co-transcriptional splicing and a maturation of the mRNA that then gets exported, fully spliced, to the cytoplasm where translation begins. So when you now sequence those RNA reads, you will see that they fall on top of the protein coding exons. That's because in these experiments, we're looking at cytoplasmic RNA, okay? In some cases, you might see some reads in the intron, and you might be wondering, what is happening here? Could it be that this RNA molecule was not fully spliced? Could it be that there's alternative splicing event, and so on and so forth? So that's the kind of thing that you should worry about. But basically, if you look at now individual cells here, every line is a different cell. And then here, every line is 10,000 cells, okay? And what you can see is that the individual cells, when sequenced to sufficient depth with many of these early approaches, you can basically see that they sometimes match exactly the 10,000 profile. But other times, what you see is that the bulk sequencing of, say, 10,000 cells is dramatically different from some cells that just simply have no expression for that gene. Everybody with me here? So that, I hope, gives you an appreciation for these rare events that sometimes are happening. Basically, this gene is only expressed in a subset of cells. And again, when you look at imaging, you see the same thing reflected there. This is not just a technical artifact. For those genes that are uniformly expressed across cells, when you stain them, you see that they are indeed found in every one of those cells. But those that are only there in a subset of cells are indeed only there in subsets of cells when you look at imaging. And those that are only in a handful are, again, only appearing in a handful. So that basically should reassure you that this is not just about dropout events, that this is really biological variability. You could call it stochasticity, or you could just simply call it variability. Stochasticity suggests that there's some kind of random process. And yeah, some of it might be random. But what do you attribute to randomness? Is it that the receptor bound and then the cell randomly chose not to respond? Or is it stochasticity because that ligand didn't actually bind the receptor in some of those cells, and then they responded in a completely deterministic way to otherwise probabilistic environmental factors? Is everybody with me here? But ultimately, what you need to understand is that the variability is there. Much of the time, it is actually biological, not technical, but some of the time it will be technical. Some of the time it will be stochastic because of the way that the environment works. Otherwise, it will be stochastic just based on the way that the cells respond to a deterministic event. And of course, most of the time, it's going to be all of the above. Everybody with me here? Awesome. Who feels that they're learning? Yes? Awesome. Good. Again, you can kind of start asking about intercellular and variability between different cells, and you can start getting a lot of insight as to what are some of those most variable or non-variable genes. Okay, so now let's see how we went from capturing individual cells in some of the earliest single cell papers, or 10 cells or 100 cells, to capturing millions of cells in the span of less than a decade, which is mind-boggling. Okay. So what are the key technologies that gave rise to this dramatic innovation? So again, if you look at the methods for isolating single cells, you could just simply pipette by eye individual cells and put them into 96 well plates and then sequence each of the wells as if it was a sample from a different tissue or a different individual. You could basically use capillary pipettes under the microscope to be able to sort of capture those better. You can use this FACS sorting, that is throwing a laser that can detect the presence of cell surface markers or other protein markers in a collection of cells. And as every cell travels through that tube, you can basically make a decision to keep it in the tube or outside based on this by applying a current or a magnetic field or you name it. And that allows you to now capture a particular cell type that you might care about. For example, you might care about activated T cells. And there's a marker of activation that you can then sort for and gather them all together. Everybody with me here? You could also use laser capture microdissection, that is basically looking at a tissue and then capturing a tiny little part of that tissue, cutting it with a laser. And one of the most widely used technologies is the microfluidic technology that I mentioned earlier, where you basically have cells from your suspension coming in one microfluidic tube. Then you have these individual beads that capture the barcodes that have, you know, again, thousands or millions of barcodes that all have the same code, if you wish. Let's say, you know, AGGTTCA for some cell, and then TGACCGG for another cell. And then every time you see the exact same barcode, you know that it was the same capsule and therefore most likely the same cell. And then by adding these capsules with oil, you're using the physics properties where these material and the oil are actually, the lysis buffer and the oil are not mixing, therefore causing this lysis buffer to sort of clump inside one of these chambers, one of these bubbles, that basically becomes your reaction chamber. Everybody with me here? And then there's, you know, other technologies. I'm going to talk about some of those. Okay. So anyway, the key idea is that the overall methodology is that you first isolate cells, then you amplify the RNA, and you can, you know, choose, for example, 2 populations of cells, and you can use microfluidics, and then you effectively, at the end, do some kind of measuring or counting of RNA molecules. And you can do that from full-length sequencing, which basically will then give you the splicing patterns for each of those RNAs. Or you can use that with what we call digital gene expression analysis, that effectively only looks at the tail end of every RNA molecule. Why the tail end? Because when we sequence, we use a primer that initiates the polymerase reaction through which we do the sequencing. And that primer, for a lot of eukaryotic genes, can be simply TTTT, because these genes finish with AAAA. So there's a polyadenylation that basically adds a bunch of A's at the end of mRNA transcripts. And then if you have the TTTT primer, that allows you to now start sequencing the tail end of these genes, and therefore capturing the number of times that mRNA was expressed, but not capturing the specific splicing pattern under which it was expressed. Everybody with me here? Who's following so far? Give me a 5, 4, 3, 2, 1. Beautiful. That's awesome. Great. All right. So again, there's many different technologies for single cell RNA sequencing. There's you know, different amounts of cells that are being captured. There's different costs. There's different sensitivity, and there's different cell capture methods. And you know, you can kind of look at this extraordinary diversity. And you can see that for some methods, you capture 50 to 100 cells, or, you know, 500 to 2,000, 5,000 to 10,000. Then there's a cost per cell So basically, when you're doing this traditional every cell is a well approach, you basically pay about $3 to $6 per cell, which sounds ridiculously cheap, unless you start caring about millions of cells, and then it becomes expensive. There's pooled approaches that are basically looking at, again, $3 to $6 per well. And then there's these microfluidics-based, individual cell approaches where you basically have, 5 cents to the cell, which again, sounds crazy. Okay, so what are the key technologies surrounding those? Again, as I mentioned earlier, you can basically use this laser shooting and then these flowing of cells to basically choose individual cells based on these multispectral detectors, and then put the cells of interest to the plate. There's these microfluidic chambers that I mentioned earlier, where you're basically capturing, encapsulating each cell in a well. And then that well, a microreactor, which is basically just a bubble, you didn't have to pay with plastic for that chamber, has a bead that then has all of the same barcode on that bead. And that barcode then gets attached to all of the RNA molecules of that cell. So you basically capture the cell inside the bubble, and then you lyse the cell. You release all of the mRNA molecules, and all of these mRNA molecules get the same tag, okay, the same barcode. So this is what the bead looks like. You have a PCR handle, you have a cell-specific barcode, and then you have something that we call a unique molecular identifier, a UMI. What is that? That is basically just a random chunk of nucleotides, where you basically synthesize these nucleotides to just be random. It could be 4, it could be 8 nucleotides, which are basically just N, which basically means any nucleotide. Why is that important? Because then you know that you're sequencing the exact same molecule that was mistakenly amplified 1,000 times. So therefore, if you see the same unique molecular identifier 100 times, it means that that's exactly the same RNA molecule that gets sequenced. And therefore, it's an artifact. You shouldn't say, ooh, there's 1,000 copies of that gene. Everybody with me here? So the UMI is usually a measure of uniqueness. And that's basically what we're going to, when we say, how many cells did I capture? You'll say, well, how many cells, that's the number of barcodes. How many unique RNA molecules? That's the UMI. Okay? Everybody with me here? Awesome. So then the coolest part about this is that after I've done this step of basically in my bubble tagging every RNA that came from that cell with the same barcode, I can then blend all of the bubbles together and do one sequencing reaction. So basically, this entire process is just to tag all of the RNAs that came from the same cell or nucleus. And then what I'm actually sequencing is just a bulk sample. It's the same thing as I did before. Okay? Only I did this magic to basically tag my RNAs based on where they came from. 5, 4, 3, 2, 1, who's with me on this one? Yes? Beautiful. Okay. So that's that. And then in the middle, you have this very cool technology that I will explain in the next slide, which is that there's another way to do this, which is you have your, let's say, million cells. And in the first round, you split them into 100 wells. And all of the 100 in this, all of the cells in the blue well, I just color blue, with a barcode that's like, I don't know, 6 nucleotides, for example. Or however many nucleotides I need to distinguish, let's say, 100 of them, okay? Whatever log4 of 100 is. You then blend all of your cells together, but now they've been colored blue or red or orange or green or yellow, etc. Everybody with me here? Because they have a unique barcode added to every RNA of that cell. You then blend them all together and split them again. You now add a second barcode on top of the first one. And this could be, pink or yellow or green, or you name it, some colors that you did not reuse before. And because this is digital, it's RNA, so you're adding individual barcodes, then these can be completely different codes from that one. Everybody with me here? And then you blend them again, and then you add a third barcode. So by the end of the exercise, every RNA in every cell has gotten 3 different barcodes. One from the first reaction, one from the second reaction, one from the third reaction. Round 1, round 2, round 3. If I have 100 wells here and 100 wells there and 100 wells there, the chance that 2 of them, that 2 mRNAs will have exactly the same barcode, but went through different cells is effectively nil. Basically 1 in 100 times 100 times 100, that's 1,000,000, okay? So I will basically have 1,000,000 distinct possible paths through the 3 rounds of wells and 3 rounds of barcoding. So then I can sequence my entire thing and effectively call all of the RNAs that have exactly the same 3 barcodes as having gone through the same 3 wells because they were attached to the same cell. Who thinks this is cool? Yes? Awesome. Good. And who's with me? 5, 4, 3, 2, 1. Yes? Awesome. Beautiful. Okay. So that's basically the, the third technology. So the second technology is by far the most common. And then, this is the one where you're basically adding these barcodes and then you're dissociating the cells and then sequencing the whole reaction. But this is, I think, just very, very cool as well. And again, it sort of teaches you different ways of combining. Okay. Everybody with me on technologies? All right, let's now talk about biology. So basically, again, there's 3 parts to the lecture. One is the technological part, one is the biological part, and one is the computational part, okay? So we talked about the technological part. Let's now dive into the biological part. What do we actually do when we have tons of single cell data, okay? And this is a lot of research that happens in my lab, and it's kind of fun because, I get to teach about stuff that didn't exist a year or 2 or 3 ago because we just built it. So let's see, what do we do? So basically, what's the goal of our lab? The goal of our lab is to understand the circuitry underlying disease, to basically look at how are genetic differences or environmental differences leading to molecular differences that then manifest in disease states. Those molecular differences can be at the RNA level or at the epigenome level, and we're going to talk about epigenomics next week. But then we profile massive numbers of both healthy and disease samples with single cell profiling. And then we carry out all of the stuff that I'm going to show you in the next section to find what are the driver genes, regulatory regions, regulators, cell types, pathways, processes, etc. And then we infer from this analysis causality, because genetics gives us causality, because we know that this genetic variant leads to differences in a causal fashion rather than those differences lead to the genetic variant, okay? Because you inherit those from your parents, and those you basically measure after a lot of environmental exposures. It's very unlikely that this is in fact causing the genetic differences, much more likely that the genetic differences are causing whatever differences you see. But of course, the genetic differences could be then, I don't know, causing you to eat a lot of McDonald's, and then the differences are not directly linked to that genetic difference. They're linked to the fact that you had an unhealthy lifestyle. Okay. Unless you're having salads at McDonald's, which is also great. We then try to infer these causal paths by understanding how are these genetic variants acting at the molecular level to then figure out where to intervene. Okay. So what I'm going to tell you now is some of the lessons we're learning for applying this to a lot of disease samples. So we have set up a large number of amazing collaborations, applied for dozens of grants from the NIH to fund each of those collaborations. And we're basically studying dozens of different traits at single cell resolution. This takes an enormous amount of coordination across doctors who are providing the samples, extraordinarily generous patients who are sharing, their samples post-mortem, and their families, of course, amazing heroes of this research. All of the technicians in the lab who are basically gathering these samples, sorting them, profiling them, then all of the experimental, extraordinary technical expertise by the experimental members of our team that are basically then dissociating, carrying out all those steps that we talked about earlier, pushing very often new technologies forward to make all of this possible. And then, of course, the computational analysts that then come in and analyze these data. And of course, the whole team together working to analyze, interpret the results. Okay, so that's, it takes a village basically to create any one of these studies. But what we basically do is that for Alzheimer's disease or for frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in the context of Alzheimer's, psychosis in schizophrenia, bipolar disorder, Down syndrome, autism, depression, suicide, PTSD, aging, for all of these differences between controls and cases, we basically then profile dozens of cell types across tens of millions of cells, very often in multiple regions of the brain, very often also in other tissues outside the brain. And then most of the time, we do RNA profiling through single-cell transcriptomics, which we're talking about mostly today. And in some cases, also DNA accessibility, which we're going to talk about much more next week. Everybody with me here? So then what do we find? So in one of the first studies in the postmortem human brain and the first study of Alzheimer's at single-cell resolution, we basically gathered 80,000 cells from 48 individuals. And this was not the beginning, but the middle of a very long and extraordinary collaboration with Li Hui Tsai and her lab. She's the director of the Picard Institute for Learning and Memory. Hans-Reddy Mattis led a lot of these studies, both experimentally and lately computationally. And, we've been, releasing a series of papers. But that was the first paper by Mattis, José David de Wilderijn, who basically now has his own lab in Italy. Hans-Reddy Mattis has his own lab at UPIT. And what we basically looked at is, what does this all mean? What are all these cells doing? Okay, so I'm going to show you a lot of the computational steps to figure this out. But very briefly, the first step is clean up the data. And I'm going to tell you some stuff about that. What does clean up the data mean? That basically means find metrics of quality that allow you to throw away individual cells, or throw away a whole experiment, or throw away individual genes that might be misbehaving, and so on and so forth. After you've done that, you basically have this expression matrix that I showed you in the last lecture. You basically have a matrix of a lot of genes and a lot of cells. Everybody with me here? The next step after that is to cluster these cells together to basically figure out what are distinct cell types, if you wish. Okay? And this can be kind of challenging. In some cases, why would you call this whole thing oligodendrocyte progenitor cells and not, a subset of those? Okay? In other cases, you have excitatory neurons that are sort of shifting across a whole gradient of layer 1, 2, 3, 4, 5 of the cortex, which actually have distinct expression patterns, but they're more of a continuum than discrete cell types. So calling individual cell types from this data can sometimes be difficult. Okay, so this is the first step, basically clustering to identify what are the major drivers of variation. And that's a form of unsupervised learning, as we talked about in the last lecture. So you've now clustered your cells, and at some point, you're going to call individual clusters, and you're going to give them a name. And you're going to say, well, these cells here that have the same barcode cluster together, and I'm going to guess that they they have a particular identity. How? By looking at what are the genes that are the most expressed in those cells. And again, we're not showing up here from, the Stone Age. We're showing up here with decades of research of extraordinary scientists minutely studying the expression of individual genes in, cells that they studied under microscope. Individual cells have always been around, and they've always been individually studied before these massive scale methodologies. Okay, so the difference here is that we're looking at 2 million cells, whereas the previous studies were looking at 20 cells or 100 cells. Everybody with me here? So with these 84,000 cells from 48 individuals, we basically asked, how does the expression pattern of those cells correlate with the phenotype of those individuals? That's the first thing you want to ask. So those 48 individuals are, shown here as different columns. And every person has some phenotype associated with that person. Some people, we're calling them controls or non-Alzheimer's. Why? Because if you look at the phenotypic signatures of Alzheimer's at the pathology level, they don't have them. Neurofibrillary tangles, NFT, pathology, neuritic plaques, amyloid beta, and so on and so forth. You see, these are very low value here, but very high value here. Is everybody with me here? Awesome. Then you can also look at signatures of Parkinsonism and signatures of cognitive decline. And you can see here that the global cognition for those individuals seems to be high, except for this one person that shows some signs of Parkinsonism, perhaps. And then the individuals here have, in general, very strong cognitive decline and a lot of pathology. And then there's another group here that is somewhere in the middle. So we call this mild cognitive impairment, or MCI, or mild AD. And we call those AD, or severe AD, and those non-AD. Everybody with me here? And then we basically ask, what is different between those people? And we can basically now start studying individual genes that are differentially expressed between cases and controls. So now that you have all of these data sets, you can now ask, as a group, are the cells that are coming from Alzheimer's individuals showing higher expression, for example, for these genes than the cells coming from non-Alzheimer's individuals? Everybody with me here? Now let's talk about statistics. What statistical test would you use? You basically have, 20,000 cells from advanced AD and 20,000 cells from non-AD individuals. What can you use? You can basically look at the distribution of values for those cells versus those cells. Everybody with me here? Seems like a reasonable approach. Problem is that this assumes that every cell was sampled in isolation. When in fact, what's happening is that you don't have just 1,000 cells or 20,000 cells from control individuals. No, we have 20,000 cells that came from 24 control individuals. So the sampling process from which you obtain those cells is actually a little more complex. It's a hierarchical process. First, you sample from a distribution of non-Alzheimer's individuals, a person, and then you sample cells from that person. And of course, not every non-Alzheimer's individual is the same. And not every Alzheimer's individual is the same. So you don't have a gradient of cells, you have 20 samples of cells. Everybody with me here? So now, as you start thinking about differentially expressed genes, for bulk data, it's super simple because you have one measurement per group. With single cell data, it's more complex because you have 1,000 measurements for each of 20 individuals rather than 20,000 independent measurements. Everybody with me here? So there's three categories of differentially expressed gene analysis methodologies. One is, let's treat every cell in isolation. And that one has a problem of you think that you have more samples than you do because you didn't sample 20,000 cells, you sampled 20 groups of 1,000 cells. Okay, so then you're overestimating the confidence of your result because your variance is artificially lower because you've only truly sampled 20 people. Everybody with me here? So that's one approach, the cell-centered approach. At the other side, you can basically say, oh, things were so wonderful when we're doing bulk analysis. So I'm going to use the same statistics. I'm going to take all 1,000 cells from each of my 20 people, average them, and now that's great. I have one measurement for that person. Okay? This is perfectly statistically sound. Is it using the data to its full capacity? No. No, it's averaging that variability. And that variability is important because if I take 1,000 cells and I see that they all have exactly the same value, and then for those 20 Alzheimer's individuals, my 1,000 cells have exactly the same value, the statistical difference between those is enormous. But I've lost that because I've averaged them. If by contrast, those 20 samples, each of 1,000 cells, have enormous variance, and I look at the control individuals and they also have enormous variance, and yes, their means are off by 1, but their variances are 20 each, I really don't have any power. Everybody with me here? So basically, this approach of treating every sample as if it was bulk is very appealing, very simple, but it throws away a lot of the statistical power you have. And in the middle is a mixture model that basically says, I have a hierarchical process where in the first level of the hierarchy, I'm sampling individuals, and in the second level of the hierarchy, I'm sampling cells from those individuals. And those methods are much more powerful statistically, but they're also much more complex. A, they take more time. B, they make more assumptions about the sampling process. And those assumptions can in fact be sometimes leading you to the wrong result if your methods are not sound. Is everybody with me here? So again, there's cell-centric approach, there's pseudo-bulk approach, where you're basically pseudo-bulking, you're basically averaging all of the cells from the same person. And then there's this mixture model, hierarchical type approach. Everybody with me here? Who feels that they're learning stuff? Yes? Awesome. Okay, so then you can do these types of analysis and then say, is there a difference in the distribution of values that are coming from, say, Alzheimer's or controlled individuals? And let's look at some of the biological findings. One of the things that we found is that if you look at differentially expressed genes between early and control in these two groups, or between late and control, then what you basically see is that in late stage Alzheimer's, there's a lot more changes, and many of them are shared between the cell types. Whereas in early Alzheimer's, you basically see much more cell type specific alterations. Is everybody with me here? So that's one of the things that you can find out. You can also ask, how does sex, biological sex, play into the equation? And what you see is that, for example, for non-Alzheimer's individuals, you find thousands of differences between excitatory neurons from male versus female individuals. And you can do the same thing for Alzheimer's individuals. And you find that the Alzheimer's cases versus controls, the Alzheimer's males versus females have many, many more differences in their expression patterns. So that basically says that maybe we shouldn't be thinking of just one uniform sample of male and female individuals. Maybe we should be thinking of sex as a biological variable and sort of how that is affecting. Similarly, you can ask about the age of those individuals. You can basically say, oh, well, maybe some of these individuals were, 99 years old and others were 89 years old. And maybe expression changes with age anyway. And maybe some of the differences that I'm finding are basically due to the age differences rather than the Alzheimer's case control differences. Is everybody with me here? Again, I'm not here to tell you all the answers. I'm here to tell you how to think about all of these questions. Sounds good? Again, you can do this with one brain region, with prefrontal cortex. But then again, when you look at how Alzheimer's progresses, what you see is that there's a stereotypical progression. It starts with something much closer to your spinal cord at the base of the brain, which is basically the locus coeruleus, and that's sort of the place that starts showing the differences first. Then the second place is the entorhinal cortex. And from there, you start seeing changes in the hippocampus. And then you start seeing changes in other subcortical regions, and eventually start seeing changes in the cortical region. So we basically said, okay, can we now study the differences that are happening in different regions of the brain between case and controlled individuals? So we did exactly that. We basically said, can I look at the entorhinal cortex? Can I then look at the hippocampus, the thalamus, the mammillary body, and then some of the cortical regions, the neocortical regions, the angular gyrus, the mid-temporal cortex, and the prefrontal cortex? Everybody with me here? What's the first thing that happens? The first thing that happens is that we go from having, one class, if you wish, of excitatory neurons, spread into different layers. From that to EEG, there's many, many different types of excitatory neurons. And if you look at the regions of the brain from which they're coming, you basically see that they're actually showing dramatic differences between these different brain regions, that some brain regions are showing only those classes of excitatory neurons. And then others are showing, much more diversity. The second thing you're noticing is that it's not just excitatory neurons and inhibitory neurons that vary between different brain regions, but it's also astrocytes, oligodendrocytes, and so on and so forth. Basically here, I'm coloring those cells by the brain region. And you can see here that there's a class of astrocytes that specifically comes from, different brain regions than the other ones, okay? So that's the first thing. The second is, okay, we now need to start annotating those additional classes of cell types. And we basically now have 30 excitatory neurons, which I'm showing here, and 23 inhibitory neurons. And for every one of those, you want to know what are the distinguishing features of those neurons? What are the marker genes, if you wish? And then you're finding, again, dramatic variability between regions. And also by having many, many more cells, we're now talking about not 84,000 cells anymore, but 1.6 million cells. And of course, technology has also improved from 2019, where we had the previous study, and 2024, where we just published this study. You basically, and this just came out in Nature a few weeks ago. So you can also start sorting the cells based on how Alzheimer's-like they are. And you can start painting a progression of cells over time. And that's one of the things that you can do. We can also do a lot of spatial analysis. I'm going to skip that for now. And you can also say, okay, great. I can study individual genes for how they're changing expression. And then I'm doing 20,000 independent tests for every one of those genes about what changes in Alzheimer's disease. For any one of those genes, I might not be very confident. But if I start seeing multiple genes in the same pathway, then I might start gaining some confidence. Is everybody with me here? But what if, instead of analyzing the data one gene at a time and after the fact asking, hey, are those multiple independently measured genes changing in the same pathway? What if instead I can analyze my data into groups? What if I could look for this giant matrix of genes by cells and then cluster that matrix, decompose that matrix using singular value decomposition? And I can basically say, okay, can I think about all of these single cells and all of these genes as being sampled from a lower dimensional space, where instead of having every gene acting in isolation, I can think of eigengenes or modules of genes that are acting together. Is everybody with me here? So then I can basically say, great, can I group all of these genes together into these modules and then start asking about, first of all, what are the genes that were grouped together into modules? And I should say that your TA here, Benjamin James, who's very busily finishing preparing and simplifying the PSET that turned out to be a little too complicated, was in fact one of the leads for this analysis, along with Carlos Bush, who is now a postdoc over at the Harvard Medical School. Okay. So basically what Benjamin and Carlos did is that they studied what are these modules actually meaning. And you can take this matrix and look at just clusters, or you can take this matrix and start asking what is the correlation between every pair of genes. And the strength of that correlation between every pair of genes can give you modules of genes. It gives you a network of genes. And therefore now you can visualize every single gene, not just if it's in a cluster or not, but where it is connected in that network of gene-gene correlations, if you wish. Is everybody with me here? And you can use that to now start studying how modules of genes are changing in a coordinated fashion across different cell types and in relation with different pathological signatures associated with Alzheimer's disease. You can say, what are the modules that are changing with cognitive impairment? Or those that change with diffuse plaques or neuritic plaques or neurofibrillary tangles or a combined NIA-Reagan score for Alzheimer's pathology. Yes?
I have a question. Why are we doing this across clusters of genes and not just individual genes? Like, what's the benefit of doing this in a cluster?
The benefit of doing it in a cluster is that you have a more robust measurement, because when you're measuring individual genes in individual cells, the sample is just too small. If one gene is expressed at 4 molecules per cell, and you're measuring individual cells, the measurements themselves are going to be very noisy. If instead you say, well, I'm not just looking at one RNA molecule, but I'm looking at a dozen RNA molecules that are co-vary, then it gives you more robustness. Does that make sense?
How does analysis compare to
With pooled RNA-seq?
bulk?
Oh, with bulk. So the first thing that you lose in bulk is the cell types. So basically, instead of saying, oh, it's this particular subclass of microglia, then you'd be like, how is this gene changing between Alzheimer's and controls across all of the cell types averaged together? Now, with Alzheimer's, you're losing neurons. So it will look like, oh, wow, all of these genes are decreasing in expression. But those are the genes that are expressed in neurons. What's happening is that you're sampling, let's say, 40 neurons per experiment in this person, and in sort of, let's say, 4,000 neurons in a control individual, but only 2,000 neurons in a Alzheimer's individual, because they've actually lost those cells. So then it will look like you have thousands of genes that are decreasing in expression. Does that make sense? Good. So basically what single cell analysis allows you to do is basically say, what are the expression changes that are happening within every one of those buckets of different cell types? That's the first level. And that you can do with even pseudobulk analysis. You can basically take all of the cells, dissociate them, measure 10,000 cells per person, and then simply condense that into 7 measurements. As if you had sorted excitatory neurons, you had sorted inhibitory neurons, you had sorted astrocyte, sorted, microglia, sorted vascular cells, and then you basically did a bulk experiment in each one of those. That's fine too, and you can do that, and that's sort of the pseudobulk analysis that we're describing there. Make sense? Great. These are all fantastic questions, so keep them coming. Any other questions? Raise your hands. Doesn't need to be fantastic. I'll try to make the answer fantastic.
I guess how consistent are the clusters with different dimensionality reduction methods?
Yeah.
And how do you actually validate this? Like,
Yeah.
who's half a connection?
Yeah. That's a great question. So basically, we've now gone from individual genes that I could just name, it's this gene, to modules. Like, module 13, what does that mean to you? And, if I run the analysis again, will there be a module 13 again? Will that module 13 be slightly different the next time around? So this is a fantastic question. And again, there's a lot of thought about exact reproducibility of the results. My take on this is that exact reproducibility is almost a red herring. And the reason for that is that if I were to do the experiment again and I would have sampled slightly different individuals, what I care more about is the reproducibility across not just, hey, can I run it on a different computer, but can I run it with a different set of individuals or a different study or a different cohort and so on and so forth. And you can ask the question of robustness to some of those decisions. For example, if I subsample cells from the same people, or if I subsample individuals and take all of the cells from those people, or if I do both, if I randomly subsample individuals and I randomly subsample cells, or if I split the cells into 2 groups, I basically artificially pretend that I have a replicate experiment, or I basically take half the cells from this person and half the cells from the same person, but different halves, and then I pretend that I have a replicate. Is that a true replicate? The answer is absolutely not, because in carrying out the experiment, I cut a particular region of that brain of that person, of that donor. And that region might be slightly different from the region that I did for the next experiment. So what's extraordinary with this cohort is that there are 2 teams that carried out a very similar analysis. Phil de Jager, a close friend and collaborator, who basically was initially at the Broad and has now gone off to Columbia, who basically profiled 200 of these individuals separately. And they did 2 separate replicates of the same, for each individual, for those same 200 people. So we basically have something like 430 individuals for our cohort, and something like, I don't know, another 400 individuals from their cohort, 200 of which are the same. So we now have 3 replicates, 2 biological replicates from the same research group, and 1 third replicate from our group for the same sample. An awesome final project would be to say, hey, how do these relate to each other? We've gone and parsed all of the data and prepared all of this, but no one in my group is working on this. So hey, and in fact, as we start talking about projects tomorrow, we have tons of data that is even unpublished. So if any of you guys are interested in sort of either diving into some of these massive data sets or taking on completely unpublished data sets that are coming out of the oven, in a couple of days, you can. And again, like, the beauty of teaching this course is that this is not like 1906 physics. This is like 2024 computational biology, and this field is changing dramatically, by the, week. So anyway, great question. How do these change between replicates? How do these change? If I use a slightly different version of the software, will the results change? Yes, they will change. Will that matter? We'll find out, right? Like, that's what this is all about. And I'm so excited about, seeing how many of these results will hold the test of time. But if you wait until you've done the same experiment 7 times before publishing it, as if we were waiting to sort of until the technology is fully mature to sequence the human genome, I don't know, we'd still be in the Stone Age wondering if we should be using fire or not, because fire can be dangerous, right? So basically, I feel that, time will tell. We publish a lot of hypotheses out there. Some of these hypotheses are replicated independently by other teams. When you compare the Phil Jaeger papers and the Ty Kelly's papers, you basically see that, a lot of the similar pathways are in fact changed. And that gives us some confidence. And, the further we go into science, the more confidence we'll have for some hypotheses. And then some other hypotheses will realize we were wrong. And then we'll just, adjust, change course, and move on. Does that make sense? Anyway, awesome questions. And I'm totally enjoying answering these questions, as you can tell. So basically, keep them coming. Raise your hands. Doesn't need to be, oh, I don't understand X. It could be like, hey, I'm wondering about Y. Who feels that they're learning a lot? Raise your hands. Yes? Awesome. Good. And who's excited about this whole field? It's amazing, right? Like so much stuff is happening. All right. In the series of analysis that you guys can do for your projects, yes, of course, you can study modules, you can study spatial transcriptomics, you can study epigenomics. Here's a very cool and very crazy kind of analysis. We basically said, what is the correlation in the way that different cell types are changing between individuals across brain regions? And what we found is that, for example, the subiculum in the entorhinal cortex was in fact, highly correlated with the corresponding, first column here, which is the, AGL, whatever neurons, okay? So basically, what that teaches us is how the different regions of, say, the hippocampus and the entorhinal cortex are co-varying across individuals. Why is that exciting? Because it allows you to now say that, oh, across my 24 individuals, this particular subclass of neurons was co-varying with that other subclass of neurons. And from, again, decades of extraordinary neuroscience, we know that these regions are actually connected to each other, that they're projecting into each other. So when you start thinking about how is Alzheimer's progressing in this typical pattern from the, locus coeruleus to the entorhinal cortex to the hippocampus, etc., one of the hypotheses is that pathology progresses either because of some physical damage or because of the way that the signals are altered and the neurons rely on those signals to basically stay alive and well. So basically, maybe the regions that are signaling to each other are correlated in their damage because of one of these possibilities. And that, that's another super exciting analysis that you can do. You can also start asking about how to group individuals and how to predict individuals based on their transcriptional signatures. So this is a different study in schizophrenia that was just published in Science a couple of months ago. And what we basically did there is that we asked for control individuals and for schizophrenia cases, can we predict whether a person is schizophrenia or control just based on their expression patterns? And what's really cool is that, yes, it did work for many people. That's cool. But what's even cooler is that it didn't work for other people. And for this particular person, control number 7, the algorithm said, wait, that person looks very much like a schizophrenia person, but this was a control. So what's happening? Is that person in fact the case? Could that person have been misdiagnosed? Are our methods wrong? Are our signatures invalid? Well, it turns out that this was the father of a son that was actually diagnosed with schizophrenia. Could it be that there's expression patterns that led to something inherited? That basically was, again, schizophrenia is 1 in 1,000. So the chance that that person's son is schizophrenic is actually quite small if it was completely independent. So maybe there's something going on here. Conversely, we found some schizophrenia cases that were just completely control-like in their expression patterns. Could it be that they're a different pathway that leads to schizophrenia, like symptoms, but it's actually not schizophrenia? We now have 430 individuals with postmortem single-cell data from Alzheimer's disease. One of the projects could be, hey, I want to study those individuals that look normal, but were diagnosed, and see if there's something different between them. I can basically look for either misdiagnosis or overdiagnosis or different classes of Alzheimer's, and so on and so forth. So again, there's an entire universe of data that you can do. Another analysis was looking at the upstream regulators that are upstream of all of these genes that are differentially changing, and then starting to predict what are what's common between these regulators. You can also start studying genetic effects and so on and so forth. One of the directions, and we've done this for pathogens, we've done this for psychosis, we've done this for ALS and frontotemporal dementia. This paper was just published in Cell just a few weeks ago. We're finding a lot of these pathways that are changing. Huntington's disease, this was again published in Cell last year, exactly a year ago. And this is looking at different subclasses of microglia. This is looking at vasculature. This was published in Nature a year and a half ago. Basically looking at the differences in expression in the vascular cells. So vascular cells are 0.3% of the data that we have. And in the 0.3% of the data, you can basically find a gradient of gene expression patterns between the vascular cells that are sitting on the venule versus the arterial part of the vasculature. And then seeing how these change in Alzheimer's, we just published in Nature Neuroscience a little bit later. And this was also published in Cell about a year ago. This is looking at single cell accessibility and epigenomic differences in Alzheimer's. This is another type of analysis that you can do, which is basically asking, can we infer from the same experiments the number of mutations that are there between the reference DNA and the sequenced RNA? Those mutations could correspond to somatic changes that are at the DNA level, or they could correspond to damage that happens at the RNA after being expressed. But what's really interesting is that if you look at Alzheimer's versus control individuals, those that actually have dementia are basically showing more mutational burden, if you wish, at the RNA level than the individuals that don't have diagnosis of dementia. And that burden is happening very specifically in a subset of cells that we call de-identified cells or senescent cells. So basically, you can actually use the RNA sequence because it's sequencing, to infer something about somatic mutations either at the DNA level or at the RNA level. What's really interesting is that those individuals that show more somatic mutations are also the ones that are showing more epigenome erosion. And that's basically when the entire global epigenome is just like flattening out instead of having clear active regions and clear repressed regions. It's sort of, diffusing out. Okay. So I want to step back and think a little bit about this diversity of analysis that you can do at the single cell level, and basically start reflecting about, basically you start with your raw data, you then have count matrices of genes by cells. And then you can carry out quality control, data correction, normalization, feature selection. And a lot of that is going to be in your problem set 1 that's going to go out on Tuesday. And you can then visualize the corresponding cells by projecting them into the lower dimensional space. And then you can start annotating them. And then you can cluster the cells together. Then you can annotate these clusters based on different names. Then you can figure out the trajectory analysis, as I mentioned earlier, by basically looking at the number of spliced versus unspliced reads for different trajectories and sort of seeing where in that trajectory something is. You can infer, of course, differential expression. You can study loss of neurons, for example, and composition differences, and so on and so forth. So one last analysis type that I want to mention is that in addition to this differential expression analysis, which basically says, I've now grouped all of the cells that I call microglia separately from all of the cells that I call excitatory neurons, or microglia subtype 12, for example. What you can do is actually start studying individual cellular neighborhoods. And brace yourselves, because this might blow your mind. What does that mean? That means that I'm not going to look at discrete cell types anymore. I'm not just going to simply cut here and say all of that is excitatory layer 5 or 6, okay? Instead, I'm going to say within astrocytes, I'm going to study the neighborhood of every cell, the transcriptional neighborhood of every cell. This is not spatial neighborhoods. This is transcriptional neighborhoods. This is not XY coordinates of a two-dimensional plane that I'm looking at the cells. This is some lower dimensional projection of the transcriptional similarity between cells. So the proximity of those cells reflects how close are they transcription. Everybody with me here? And now, because we have 430 individuals, you can basically look at the phenotypes of those individuals and project them onto those cells, right? This is what we call cell-projected phenotypes. So we basically project AD versus non-AD on those cells. What do you see? Some cells are colored blue and other cells are colored red. Some neighborhoods are colored blue and other neighborhoods are colored red. That basically means that within the cell types and the subtypes, there are now additional infinite resolution subdivisions that are basically telling you that Alzheimer's cells are moving towards here and control cells are moving towards there. Everybody with me here? Why is that exciting? Because I can now annotate that neighborhood as more control-like and annotate that neighborhood as more Alzheimer's-like. And what's really cool is that I can take an individual and forget about their phenotype and then go back and ask, does that person have their microglial cells in more Alzheimer's-like states or more control-like states? And the same person might have more Alzheimer's-like microglia, but more control-like oligodendrocytes. Are you with me here? So instead of having the phenotype be something that I measure once and I'm done, you now start thinking about phenotype at the level of individual cells. And that allows you to now completely increase the resolution of your analysis, because you can then go back and re-annotate your individuals by basically saying how Alzheimer's-like is every person in their oligodendrocytes. And that gives you a measure of oligodendrocyte pathology that actually correlates with additional measurements that you can do at the cellular level for the same people. So through the single cell analysis, you can do an extraordinary diversity of different types of annotations. And you can now go from the neighborhoods to paint every cell, Alzheimer's-like or not, and to paint every individual, and the sky's the limit. Okay? Needless to say that I'm excited about this field. I'm excited about where this is all heading, and I'm really excited to hear your ideas about final projects tomorrow. So we're going to meet in this room on Friday at 3 p.m. for recitation, as always. But instead of going over a problem set or additional material, what we're going to do is spend the entire time going over your project. So if and your ideas and sort of what you want to do. Okay. So if you have ideas for projects, now's a good time to start thinking about them. Read ahead from the material. We've only had 2 lectures, so it's very difficult to think about projects, but think about the things that you care about. And a lot of you guys have already submitted really awesome and very cool ideas. Tomorrow, we're going to form sub-teams, and then we're going to start thinking about the types of projects we can do in different parts. And we start thinking about data sets from papers, from unpublished collaborations that you might have, from data that we have in our group, from recently published papers, from our group or other groups, and so forth. Everybody's excited? Who has not yet submitted their first day survey? Raise your hands. Okay, awesome. Who has? Nice, awesome, great. So please submit if you haven't already. Just sent you an email with a link, and then see you guys tomorrow.